about a recent AgriFutures project of the effects of host pathogen genetics and nutritional resilience on chalkbrood outbreaks. So chalkbrood disease is caused by a fungal pathogen, Ascosphaera apis, and it infects honeybee larvae. When larvae eat fungal spores, these spores will germinate in the gut and the fungal mycelia start to grow and penetrate the larvae body and will eventually cover the larvae with the white mycelia, and that's where we see these typical chalkbrood mummies. When we get the black chalkbrood mummies, that's when two different mating types of the fungus have come together and have made these black reproductive spores. And it's these spores that will go on to infect new larvae. We've had chalkbrood in Australia since 1993, when it was first found in Queensland, and has pretty quickly spread to the rest of the country. And since that time, it's typically been thought of as a largely stress-related disease that's only seen in spring. But in more recent years, beekeepers, both in Australia and overseas, are reporting more serious issues with chalkbrood, and these problems also seem to be extending well beyond spring. Whether a colony becomes infected by chalkbrood and the severity of infection is going to be influenced by the interaction of different factors. So the bee's genetics will affect the larval resistance to infection, but also affects the social immunity behaviours, such as hygienic behaviour. The chalkbrood genetics will affect the virulence of the pathogen to cause infection and also to spread, with some genetic strains being more infectious than others. And then there's a range of environmental factors that are implicated in the severity of chalkbrood disease. And these include things like the weather conditions, the availability of food, and even how the hive is managed, so things like the frequency of comb replacement. So AgriFutures funded this study to look at some of these factors that could be influencing current disease outbreaks. And this is followed on from two important other AgriFutures studies. The first of these was by Ben Oldroyd and Jean Chapman at Sydney Uni, and they did a nice study looking at the mating levels of commercial queens in Australia. And one of the things they found was that we could improve mating levels of commercial queens. And there's actually a good amount of overseas literature supporting the benefits of higher mating levels for colonies. More recently, there's also been the PhD research of Jody Gertz, who's done a lot of work on chalkbrood, in particular hygienic behaviour, and also the first work identifying chalkbrood strains in Australia. And one of the interesting things from her research is that we actually have pretty good levels of hygienic behaviour in Australia, but despite this, we're still seeing these increasing problems with chalkbrood. So with the help of some New South Wales beekeepers, we visited apiaries with chalkbrood infected hives and collected samples from 32 infected and 32 healthy hives across 5 apiary sites. We collected worker bees, infected and uninfected larvae, stored pollen from the hives, and also took photos of all the frames in the brood box. And then we revisited these hives about 2 months later to see if they were still infected or not. And this let us characterise the hives as either being chronic or acutely infected. For this study, we took a mostly molecular approach to compare chalkbrood infected and healthy hives, and targeting those genetic and environmental factors that we thought were most likely to be influencing current outbreaks of chalkbrood. So for the genetic factors, we looked at the mating levels of these colonies, or in other words, their genetic diversity. We looked at the baseline immunity of the larvae, and also the strain diversity of the chalkbrood infecting those colonies. For the environmental factors, we were targeting things more associated with hive nutrition. So we looked at the levels of brood and food in the hives. We examined the diversity of pollen in those hives using metabarcoding, and we also used a quantitative PCR approach to look at the levels of contamination of that pollen with chalkbrood spores. We were also interested in whether there's differences in the gut microbiomes of adult bees in healthy and infected hives. And lastly, included levels of nosema as a potential contributing factor. And then in a separate experiment, we also took an in-hive inoculation approach to expose larvae to 3 different chalkbrood strains, and doing that to compare the virulence of those strains. So today I won't have time to cover all the things that we did in this project. I'll just be pulling out those key factors that seem to be involved with these chalkbrood infections. But do check out the full report that's on the AgriFutures website. So one of the things we found was that colony genetic diversity was an important factor for these colonies. By genotyping workers, we were able to infer the number of drones that the queen had mated with, and then we are able to use a formula to estimate an effective mating level for that queen. And this gives us a more accurate measure that accounts for paternal bias in the offspring. So queens of chalkbrood infected hives had fewer mates than healthy hives, and this was more significant when comparing the effective mating level. We know this from overseas studies that high mating levels can improve colony survival and disease outcomes. But from these studies, an effective mating level of 7 has typically been thought of as sufficiently mated. And the mating levels that we observed here were typically higher than this. So we are suggesting that perhaps this threshold is not high enough to see the benefits for chalkbrood resistance. So for instance, just taking the average effective mating level of the chalkbrood hives, which is 13, we found 71% of infected colonies were below this threshold, but only 25% of healthy hives had an effective mating level less than 13. We also found that the levels of stored pollen in hives was also a key factor for chalkbrood infection. So we had taken photos of frames in the brood box and used image analysis software to measure the area of pollen on each frame. And what we found was that hives experiencing chronic and acute chalkbrood infections had lower levels of stored pollen compared to the healthy hives, and even compared to hives with only low infections. The more heavily infected hives typically had about 1,000 square centimeters of pollen area, which is roughly 1 full side of a frame. And what this result really highlights is that importance of hive nutrition for being more resilient to disease. So hives with low pollen levels are generally less able to support the nutrition of developing larvae, and this makes them more susceptible to infection. What was interesting though is that the DNA analysis of the pollen didn't find any differences in the diversity of pollens between chalkbrood and healthy hives, even though this is something that the diversity of pollen has been something that has been found important for bee health in overseas studies. We then put this data through a statistical model to get further insight for how mating level and pollen levels might influence the infection outcome of a hive. And this model suggests that a colony is 49% more likely to be healthy for each additional drone a queen mates with, and that a colony is at least 3 times more likely to be healthy for every half frame of pollen. So if we think about some hypothetical colonies in the same environment, this model suggests that a queen that has mated with the minimum 7 drones and has half a frame of pollen in the brood box has a very high chance of being a chalkbrood hive. Whereas if the queen mated with twice as many drones and had a full frame of pollen in the brood box, then the chance of being a chalkbrood infected hive drops dramatically. Now, of course, all models are wrong, but this does help illustrate how colonies within the same apiary having different mating levels and pollen stores can have very different chalkbrood infection outcomes. For the chalkbrood genetics, we cultured isolates from 3 chalkbrood mummies per hive and genotyped them by sequencing 2 fungal genes that have been used to distinguish chalkbrood strains around the world, and also recently by Jody Gertz, as I mentioned earlier. These genes are able to split chalkbrood strains into 7 master strains labelled A to G. And what we found in these colonies was consistent with Jody's work, where strain A is definitely the most common strain in Australia, and we found strains B, F, and G in less than 20% of the samples. We also observed that colonies with higher infection levels were typically infected by multiple strains. And this could be that there's just more opportunity to detect these less common strains, but it could also be that these other strains are more likely to cause severe infections. Which brings me to the infection experiment, where we use some of our Siro hives to inoculate individual thread-eyed larvae in the hive with different chalkbrood strains, A, B, and F. And then we put those larvae back into the hive to further develop and pulled them out 4 days later to count the number of larvae that had either been hygienically removed, become chalkbrood infected, or had successfully pupated. This experiment was done in early 2020, right in between the summer bushfires with smoke blanketing Canberra and followed by COVID lockdowns a few months later. So we feel pretty lucky to have this data. What we found was that more larvae were hygienically removed when infected by strains B and F, and there was a similar trend for high larval infections for strains B and F. And putting this data through a logistic regression model suggests that larvae are more likely to become infected when exposed to strain F and strain B compared to strain A. Now, there's a few limitations with this study, but it does indicate that virulence differences between strains could also be contributing to current outbreaks. So, in summary, we found that colony genetic diversity, levels of stored pollen, and virulence differences between chalkbrood strains appear to be key factors in these chalkbrood infections and are likely interacting as well. So what does this mean for beekeepers? Well, some practical actions that are likely to help reduce the incidence and severity of chalkbrood are to increase some queen mating levels and boost pollen levels, particularly in spring. Now, improving mating levels is largely in the domain of queen breeders, and it's for them to adopt strategies that really maximize mating levels. A key part of this is likely to have greater focus on drone production and retention in hives. There's also an opportunity for greater adoption of artificial insemination as a way to ensure higher queen mating levels. But managing hive nutrition is a responsibility for all beekeepers, and it's really one of the key tools for managing hive health. And so ensuring there's sufficient pollen stores in spring, perhaps upwards of 1 full frame, and whether that's through access to natural forage or pollen supplementation. This could be a really good strategy for reducing chalkbrood outbreaks. And lastly, there's certainly more we could be doing to breed for genetic resistance to chalkbrood. So in addition to breeding for hygienic behaviour, also selecting for larval resistance to infection. So I'll leave it there. Thanks for listening today, and check out the AgriFutures website for the full report of this work and also those relevant studies that I have mentioned as well. And I'm happy to take any questions.